cute little champions. I hope that you are doing wonderful as usual. And as I said you earlier, we are going to do something about the biology students who have opted biology in KCET 2025. And this is that we are going to solve top 30 questions from each chapter that we are having. And today it's turn for principles of inheritance and variations, which you know has high weightage, right? So we cannot miss this chapter. And before I get into the questions, I want to tell you something very honestly. Listen, this is going to help you analyze your preparation. So what you can do is, whenever I show you a question, pause the video, read the question by yourself and then tally your answers with me. Do not just rely on my answers, because I want you to genuinely try. What if you're not able to do? It's okay. Try it out once and then only come to that conclusion that, okay, I'm not able to do, I need ma'am's help. Yeah, works? So let's go ahead and let's see the first question that we have over here. So in male heterogametic type of sex determination, what happens? So what do you mean by heterogametic? Heterogametic means basically it's not similar. Hetero means it's not similar. Homo means it's similar. So basically I understand that it's talking about that the male would be heterogametes for something, heterogamous for something. That is, suppose you have, I am saying that the female has 2 X chromosome and the male has 1 X and 1 Y chromosome. Okay. Now when they are going to form the gametes, only one of this guy will be going to one gamete, either this chromosome or this chromosome. But either way, if you see, it will be X only in the gametes. It will be X only. Okay. So the type of gametes that are being produced is same. So I say that this is female homogamety. This is what? Female homogamety. Both of them are same. Okay. Now over here, if I see, this chromosome can come or this chromosome can come. If in this case it is X, in other case it will be what, children? Y. So if you see the gametes which are formed, they are not similar. One is X and the other one is Y. So basically I call it male heterogamety. Fine. Works. So in male heterogametic type of sex determination, males do not produce gametes. No, children, they definitely produce gametes. Male parent produces similar gametes. No, they are not producing similar gametes. Female parent produces dissimilar gametes. No, children. Female, basically they will be showing homogamety. So male parent produce dissimilar gametes. Yes, they are showing dissimilar gametes. They are producing dissimilar gametes. So this becomes the correct answer. Okay, who all show it, children? You have in the case of human beings, Drosophila. Okay, Drosophila, they also show this. So there are different organisms. Or if you talk about insects, they also show heterogamety, but in them some things are different. Okay, they also have female as Xs, but in the case of male, they just have X. There is no Y chromosome also. So in them there is X and O type of sex determination. Okay, the male will be having sex and O. O as in there is nothing. There is nothing. There is only one single chromosome, that is this. For example, if a gamete gets this chromosome, it will be X. Okay, if a gamete gets this, there is nothing as a sex chromosome over there. There is nothing. So basically, one will be having the chromosome, other one won't be having the chromosome. So here also, if you see, there are 2 types of gametes being produced. So that is also an example of male heterogamety, which is shown in the case of insects. Works? Okay. So I hope you scored 1 mark. Sorry, basically your marks in that. As of now, I am giving you 1 mark. Going ahead. In one of the hybridization experiment, a homozygous dominant parent, homozygous, okay, homozygous as in same, dominant, for example, let's take capital T, because capital T is a dominant trait, right, for tall, and homozygous recessive parent. So homozygous recessive parent as in both of them has to be the same and they have to be recessive. So dwarf gene is recessive, so we'll put that there. Now are crossed for a trait. Plants show Mendelian inheritance pattern. So what are the things that you could find if I am crossing them? What is the thing that you could be finding? So dominant parent trait appears in F2 generation and recessive parent trait appears in F1 generation. So let's cross and see. So this is the first generation that we are having. We are going to make the first generation, right? This is the parents that we are having. What will the gametes, children? The gametes could be capital T, or it can be small t. Now this guy can fuse with this, giving capital T, small t. Again this with this, capital T, small t. Here this with this, capital T, small t. Here this with this, capital T, small t. All of them in the first generation. This is the first generation product. But if I see, all of them have a capital T and a small t. They are all heterogamous. Okay, all of them, they are heterogamous. And over here, if you see, if you see, sorry, heterozygous, not homogamous, heterozygous, and over here if you see the capital T is there, so it will be dominant over this guy. So all of them will be tall children. So in F1 generation, all are tall. The dominant trait is being experienced, is being seen. Dominant trait is being seen. Now, if I try to cross them and give F2 generation, what will be the result? Let's see that. So I'm taking two of them, capital T, small t, capital T, small t. Over here, the gametes will be either capital T or small t. Here also capital T or small t. When I fuse, I get capital T, capital T, capital T, small t, capital T, small t, and small t, small t. So if I see, this will be tall because both of them are capital T, capital genes. This will also be tall because this is dominant. This will also be tall because this is dominant. So I got 3 tall. Okay, and then there is one that is dwarf, small t, small t. That is dwarf. This is what, children? This is the F2 generation. Now let's try to solve the answer. So solve the question. Dominant parent trait appears in F2 generation and recessive parent trait appears only in F1 generation. Do you see recessive parent trait ever? No. Here you see tall, that is dominant trait. So this is wrong. Next thing, dominant parent trait appears in F1 generation and recessive parent trait appears in F2 generation. So dominant parent trait is appearing in F1 generation for sure. Okay. In F2 generation, you are also seeing the recessive parent trait. That is this, at least one in 4 out of 1. Okay. 1 out of 4, sorry. 1 out of 4 will be what? Dwarf, showing the recessive parent generation. So this is kind of correct. Let's go ahead. Dominant parent trait appears in both F1 and F2 generation and recessive parent trait appears only in F2 generation. This becomes more correct. Okay. Dominant parent trait, if you see, it is appearing in here and here, and recessive parent trait is appearing only in F2 generation. So this becomes more correct, and I'll say this is wrong now. Okay, because you have a more correct statement. Now dominant trait appears in F1 generation, okay, and recessive trait appears in F1 and F2 generation. That is wrong. So what do you have as the answer, children? This is the answer. C option is the answer for this question. Okay, and initially what do? You just read 2 options and you give tick marks to that. No, you have to read the next option also. Okay, sometimes you can see varieties of things. So this came in 2023 and I hope you got the answer. Works? Next question. Which of the following statement is correct? Female carrier for hemophilia may transmit the disease to sons. Female carrier for hemophilia. So basically, if that person is a carrier and if it's a female, so it is XX. For females, you have XX chromosome, and if that person is a carrier, then that means only one of the X is having this particular mutated gene over there. Okay. So this is the case of a carrier. Now, female carrier for hemophilia, she is a carrier for hemophilia, may transmit the disease to son. May, it's a term that is may. Can she transmit the disease to the son? Let's see how. So basically, suppose there is XY, that is a male, the partner, male partner that you are having. Okay, now suppose from here, this can be a gamete and this can be a gamete. From here, this can be a gamete and this can be a gamete. When you fuse this with this, you get XX, one of them will be having H. When you fuse this with this, you get XH, Y. Okay, so this has to be a male. Okay, next thing, when you fuse this with this, you get XX. When you fuse this with this, you get XY. This is also a male. And the question says, female carrier for hemophilia may transmit the disease to the sons. If you see over here, there is H over here, and hence, since there is only one X, okay, over here you have 2 things, 2 X's, which is why this will overshadow the effect of this, and hence she will be just a carrier. She won't be having the disease, she will be just a carrier. But over here, if you see, there is no one to overshadow the effect of this guy. Hence, this guy becomes a patient of what? Hemophilia. Whereas if you talk about this is normal. This is normal. So there is an option that is may transmit the disease. Yes, she may transmit the disease or may not. This is the option and that is correct. So first option is correct, but let's see the other things also. Thalassemia is a qualitative problem. Is thalassemia a qualitative problem? How does thalassemia occur? First thing. So thalassemia basically is a disease which is caused when your hemoglobin producing genes are affected. Okay, there are two genes. One is alpha gene and one is, there is alpha protein and there is beta protein. Okay, alpha globin chain or beta globin chain. There are two chains. So though both of these chains have to be made for making a hemoglobin. You need both alpha and you need both beta to make a unit of hemoglobin. Now for alpha, to make this particular protein, you have two genes, HbA1 and HbA2. HbA2. This is for this particular protein. For this particular protein, you have HbB. That's all. You have HbB. Okay. So, if there is, this is a gene, this is a gene, this is also a gene, and all the genes will be having their alleles. Okay. They will be having the alleles. So, if there is any mutation in one of the alleles over here, or probably both of, if there is a mutation in this particular gene, if there is a mutation, suppose there is mutation only in one gene, still there will be another gene that will be making alpha globin chain, but the problem is there will be only a little amount of alpha globin chain that will be produced. Similarly, in the alleles for this also, there are chances that only one allele is affected, another allele is still being there. So that means you are messing up with the quantity, not the quality. You are messing up with the quantity of the globin chains which are being produced over here, which is why we say thalassemia is a quantitative problem. It's not a qualitative problem, it's a quantitative problem. If, for example, both of these guys are affected, both of these genes are affected, now do you think alpha globin chain will be produced? No. That means it's a severe case of thalassemia. If only one is affected, that means it is not that severe, that person is minor. Okay, thalassemia minor. If you have both of the genes getting affected, then it is major. Okay, similarly, if you have both of the alleles for this particular gene getting affected, that means it's a major problem because beta globin chain as a whole is not being produced. But only one allele of this particular gene is affected, that means at least something is being produced. So it is a minor case. Get it? So it is not a qualitative problem, it's a quantitative problem that we have for thalassemia, and please remember it. Questions have come around it. Okay. Now change in whole set of chromosome is called aneuploidy. No children, when you have a change in whole set of chromosomes, okay, you call this polyploidy. What do you call it? You call it polyploidy, and that happens in the cases of plants usually. Aneuploidy basically means when there is a change in, when there is a gain or loss of one chromosome in a whole unit. For example, if I say I have 23 chromosome and out of this the 21st chromosome is missing, that means this is an aneuploidy condition. Okay, there is a loss. Or for example, I gained one extra chromosome, that is a gain that I am having. So over here, what do you have? You have a gain, and that is why you call it aneuploidy. But it is not a change in whole set of chromosome, no children. That is known as polyploidy, not aneuploidy. So this is also wrong. Sickle cell anemia is a quantitative problem. No, it is not a quantitative problem. It is a qualitative problem. Quantitative was this. Why do I call sickle cell anemia as a qualitative problem, my dear children? Because what you are doing in sickle cell anemia is that sickle cell anemia is also related to blood disease. It's a blood disease where your, again, beta globin chain is affected. Beta globin chain is affected, but this time the quality of beta globin chain is affected. In the beta globin chain, to make this beta globin chain, okay, you need so many, codons, you need amino acids, right? So basically a codon which codes for amino acid, that is GAG, okay, here there is a point mutation occurring, which is why it becomes GUG. And now this codon which was initially coding for glutamic acid would be now coding for valine, that's another amino acid. So basically, because of this mutation that is happening, a whole amino acid is getting changed, and one single amino acid, when it changes, obviously the whole protein also changes. So hence you are messing up with the quality of the beta globin chain which is being produced. Now this type of beta globin chain which is produced is not normal. It is not normal. Okay, this is not normal, which is why the cells which carry the hemoglobin, which has this beta globin chain, will be in sickle shape. That is why it is known as sickle cell anemia. So it's a qualitative problem, not quantitative problem. Okay, when the quantity of the genes are affected, and which is why the proteins which are being produced, that is also affected, their quantity is also affected, that is what you call as quantitative problem, which is thalassemia. Okay, and over here you are talking about qualitative problem, and this is wrong. It might be a bit confusing, but children, trust me, write it somewhere. Okay, trust me, write it somewhere that thalassemia is a quantitative problem, whereas sickle cell anemia is a qualitative. Thalassemia is a quantitative problem. Okay, it messes up with the quantity of the proteins being produced. Okay, now sickle cell anemia is a qualitative problem. Okay, you can write like that. How you can remember is sickle cell. Normal cell shape is what? Discoid shape, or basically round oval shape. But over here you are changing the shape of the cell to sickle. So that is messing up with the quality. You can remember like that. Okay. So this becomes the correct answer. This came in NEET, sorry, not NEET, KCET 2023. It happens. Okay, next question. Find the correct statement. Generally, a gene regulates a trait, but sometimes one gene has effect on multiple traits. So usually we are saying that basically there is one gene and this gene will be responsible for one particular trait. But now you are saying, but sometimes one gene, this particular one gene has effect on multiple traits. This, if for example, if there is some mutation in this particular gene, this trait will be affected, this trait will also be affected. That is kind of rare. And what do you call this as? You call these genes as pleiotropic genes, or this condition as pleiotropy. What do you call this? This is known as pleiotropy. This is correct. Now, the trait AB blood group of a man is regulated by one dominant allele and another recessive allele, hence it is codominant. So, trait AB blood group. In AB blood group, I cannot say that one is dominant, other one is recessive. No. I call that this shows codominance because both of them are equally dominant, my dear children. This is also a dominant gene, this is also a dominant one, which is why I say that is codominant. Over here it is written that one is dominant and other one is recessive, which is why I call it codominant. This is wrong, but this is correct. So what do I get? Statement 1 is correct, statement 2 is wrong. So both statements are wrong? No. Statement 1 is correct? Yes. Statement 2 is correct? No. Statement 1 and 2 are correct? No. So this becomes your answer. This came in KCET 2023. I have a hang of saying NEET. I'm so sorry for that. Okay. Now, XO type of sex determination and XY type of sex determination are the example of. I told you XO, that is there is no. Okay. There is nothing, no chromosome over here. That is XO. Okay. And XY. This is an example of what? Male heterogamety or female heterogamety. This is male heterogamety. This happens in the case of human beings, Drosophila, and this happens in the case of what? Insects. In both the cases, the females will be XX only. So, females are homozygous over here, or they are homogametic over here, or they show homogamety over here. Whatever you want to say, you can say. But over here, the males are the one that are heterogamous. So, male heterogamety is being shown over here. You see, so many questions have come around this topic, so that is why it is very much important. Okay, so this came in 2022, and children, I am telling you, this topic is important for your PU2 boards also. Now, next, example for non-Mendelian disorder. So, what do you mean by Mendelian disorder? That is the first question. What do you mean by Mendelian disorder, my children? Whenever you say there is a mutation in one particular gene happening, that is what you call as Mendelian disorder. For example, you saw sickle cell anemia, you saw thalassemia, there is some mutation in some gene. Hemophilia also, there is some mutation in one gene. Okay, cystic fibrosis, that is also something. But when the number of chromosome, okay, so there are two types of diseases. One is Mendelian and the other one is chromosomal. Chromosomal is the one where, for example, I have an extra chromosome now, which is why there will be a whole problem, or probably I do not have any other chromosome, I just have one chromosome. From 23 chromosome, I am deleting one. From 23 chromosome, I am taking one more. So, this is an extra thing that is occurring. And because of this extra chromosome, because of this loss of chromosome, I am losing all the character, I am gaining extra characters that I should not be having. So, basically, this comes under chromosomal disorders. But when you talk about, for example, in one chromosome, they have normal set of chromosome. But the problem is that in the chromosome, there is one gene and this one gene is mutated. This one gene is mutated, and whatever disease is caused by this mutation, okay, will be coming under the Mendelian disorders. So, it's talking about the genes, children. So you have certain diseases like thalassemia, like hemophilia, like cystic fibrosis, which all comes under Mendelian disorders. Okay, but chromosomal, if you see, its example is Down syndrome. You have Down syndrome, where what is happening, children? 21st chromosome, there should be a pair of chromosome, right? It should be like this, but you have an extra chromosome over here. So 21st chromosome is extra. You call it trisomy. Trisomy as in 3 of them are there now. So usually have a pair of chromosome. Okay, 23 pair we say, right? 23. So 21st pair has one more chromosome over here, extra. So which is why you call it trisomy of chromosome. That is known as Down syndrome. Apart from that, you have Klinefelter syndrome. Apart from that, you have Turner syndrome. In Turner syndrome, you do not have one X chromosome. In Klinefelter syndrome, you have one extra chromosome. You get it? So basically, you're messing up with the chromosome number. So this is an example of Down syndrome. This is an example of non-Mendelian disorder, my dear children, and this came in 2022 KCET. Okay. Next question. Gynecomastia is a symptom of. What do you mean by gynecomastia? Gynecomastia basically means development of breast. Now that should not be a symptom in a case of a normal person. For example, a normal female. So this would be a symptom. Okay, development of a breast will be a problem for a person who is a male, right? And obviously this disease is known as Klinefelter syndrome, because what happens in Klinefelter syndrome is that basically, suppose this is XX chromosome and this is X and the other one is Y chromosome, right? Agreed? So over here, there's a twist that is going to happen. But just give me a second for that twist. I need to turn off the AC. Okay. So yeah, we were here. So basically, what happens is that usually to form a male, what do you need? One X chromosome from this side, okay, and one Y chromosome from this side, such that when they come together, it will be XY. But over here, there's a twist. Okay, suppose one more extra X chromosome is coming over here and one Y chromosome has come from there. So basically now what do you have? You have XXY, my dear children. There is one extra X chromosome, or probably from here only both of these X and Y came, and from here there is only one X. So anyways, you are ending up with XXY condition. So here, if you say that 46 number of chromosomes are there in normal human beings. Okay, 46 number of chromosomes are there in normal human beings, 23 pairs. Out of this 23, basically the 23rd pair is the sex chromosome. So we can say 22 pair is basically autosomes, autosomes as in the one that is not sex chromosomes. Okay, rest 23rd and 24th, basically the 23rd pair will be the sex chromosome. Okay, so wait, I don't want to confuse you. Even I will get confused. I don't want you to get confused. Listen to me carefully. You have 23 pair of chromosome, that means 46 number of chromosomes. Okay, and I am telling you, out of this 23, 22 of them are autosomes. That means they are chromosome pairs which does not have anything to do with the sex of the organism. But the 23rd one, the last one which you are having, the 23rd one, it is what? The sex chromosome. So can I say that basically if total becomes 46, can I say that I'll say 45th and 46th is sex chromosome, rest all until 44 they are all autosomes? Yes. So they will be having, in the case of Klinefelters, they will be having 44 autosomes, it's all normal, but apart from 44 autosomes, normal condition is XY like this for males. Okay, they have two more extra chromosomes, 45th and 46th chromosome. But over here, I will be finding something like this, where it is XXY. So in total, how many number of chromosomes are there? This is 44, 45th, 46th, 47. They will be having 47 number of chromosomes, my dear children. There will be one extra chromosome. So do not bring changes over here. This is fixed. 44 autosomes are fixed over here. Okay, this is the change that is coming in the sex chromosome. So bring changes over here. So they have 47 in total, and the condition will be appearing like this: 44A plus XXY. Cool. So this is Klinefelter syndrome, which usually shows all the male characteristics. Apart from that, there will be certain female characteristics that will be developing. One is gynecomastia, that is development of breast. Okay, works. This came in 2022. Okay, next, the affected male in the pedigree is symbolized by. So males are usually symbolized by this thing. Now if you want them to get affected, then you have to color it. So this will be the answer. Okay, now this shape, what is this shape means? This shape means the sex is not specified, we do not know the sex. Now here also it is colored, that means the person who is this, whoever sex is this person is affected. What is the meaning of this circle? That means it's a female. Now if I color the female, that means this female is affected. Over here I am talking about affected male, and that means this. This came in 2022. Okay. Next question, match column 1 with column 2 and find the correct answer. So with this you get a lot of things clear in your head. So I told you aneuploidy is when you will be having a loss or gain in number of chromosome. That is what you mean by aneuploidy. So I'll just go and directly mix it with this. Polyploidy means you're changing the whole set of chromosome. You're increasing the whole set of chromosome, extra whole set of chromosome is coming. That is what you mean by polyploidy. Monoploidy, the name also suggests mono. Mono means there will be a single set. You're talking about a single set of chromosomes. So monoploidy, a single set. Okay. Diploidy, diplo means 2, right? So basically diplo means 2 sets of chromosome. Get it? Easy question. Aneuploidy is basically loss or gain of a chromosome, that is shown by Klinefelter syndrome, Turner syndrome, or basically Down syndrome. Okay. And monoploidy is basically a single set of chromosome. Diploidy is 2 sets of chromosome. Polyploidy is increase in whole set of chromosome. So if you go along with that, then A is with 2, A is with 2. So this is the option, that is the only one matching. Let's see. B is with 4, yes, B is with 4. C is with 1, yes, and D is with 3. Yes. So this becomes the correct answer. This came in 2021, my dear children. Works. Next question. 10th, the genotype of a husband and a wife are IA, IB and IA, IO. Among the blood types of their children, how many different genotypes and phenotypes are possible? Let's begin that. First tell me, children, if A, B and O genus are there, okay, this is represented by IA, this is represented by IB, and this is represented by either IO or it is represented by I. Okay, is this dominant gene? If something else come, will it show up? Yes, this is a dominant one. This is also a dominant one. When both of them come together, it will be AB blood group because both of them will show codominance. O is a recessive trait. O is a recessive trait. So it won't be showing its effect until and unless it's just O there. So let's see. Over here, the parent genotype is this, IA and IB. Okay, and then you have IA and IO. Okay, what is a blood group, children? A and B both are there, so that means this person is having blood group AB. Over here the blood group will be A and O is there. A is dominant over O. You saw that O is recessive character, so it will be A blood group. That is just for you to understand things, okay? Now we are going to study. So from this parent I can either have IA or I can have IB in the gamete. Agreed? Here also I can have IA or I can have IO in the gamete. Now this gamete can fuse with this, so it will be IA, IA. What is a blood group? Both of them are A, so it will be A blood group. Now this can fuse with this, so I'll get IA and IO. Here A is dominant over O, which is why it will be A blood group again. Here I'll get this, that is IA, IB. So that means A and B both are there, they are co-dominant, so it will be AB blood group. Here I get this, that is IB and IO, right? You always write the dominant one first. Here since both are dominant, you go according to the alphabets A and B, okay? So here B is dominant over O, so I get B blood group. What are the blood groups that I can find? A blood group, AB blood group and B blood group, okay? What are the phenotypes? So these are the phenotypes. This thing is a physical appearance. Basically I can see it. Okay, this person has this blood group. So basically how many? This is A blood group, this is one thing that I can see, this is AB and this is B. 3 things I can see. 3 type of blood groups I can see. So this 3 phenotypes are possible. 3 phenotypes, as in physical appearance, okay? Even though it's not physical appearance, but still we say the physical character that we are having. Okay, so it's the 3 genotypes that we can find out, at least by blood group finding. We can find that we have this particular blood group. We cannot find the genotype, we cannot see this genes present, obviously, okay, but at least we can find the blood group, right? So if you're asked what are the genotypes that are possible, so if you see, this is one type of genotype where AA is there, then there is AO, then there is AB, then there is BO. So here none of them are same. The genotype, the genetic character is kind of different in all of them. 1, 2, 3, 4. So how many genotypes can be seen? 4 genotypes and 3 phenotypes. Let's see. 4 genotypes and 3 phenotypes. You have this as the answer. 2022 question. Works? All cool? Okay. I hope you're understanding. Now children, if you're having fun, you're finding it very easy to understand things, definitely let me know. And if you're finding it difficult, then also let me know. It's totally fine. And please take your gap and try to understand. Please go calmly, okay? Do not put too much stress. Why is it that you're not understanding Bhushan? You should be understanding it. No, you should not be doing like that. So I was supposing that person's name is Bhushan. It can be anything, okay? So please calm down and study. It's okay. You are in the learning phase. You still have time. You are trying, and that's way too good. Remember that. Now, question number 11. What is a possible blood group of children whose parents are heterozygous? Heterozygous, that means it's not the same thing. Heterozygous, that means, for example, I am saying that a blood group guy is there and he is heterozygous. That means it cannot be IA. Can it be IAIB? No, in that case, both of them would have been showing up. So it has to be IAIO. That is the only possibility that I am having. Okay, and for B blood group also it's same. Can it be IB? No, it's asking about heterogamous. They are not heterogamous, it's homogamous. Next thing, if it is IAIB, still it does not work. It should be AB blood group. It should be IBIO, my dear children. This is the thing. So first thing that you have to do is find this. How will you find this? If you read the question properly, you will be able to find it. What is the possible blood group of children whose parents are heterozygous for A and B blood group? So this is the only thing that is possible for A and B blood group. Now I am going to fuse them. Let's see. So I have IA and IO, and then I have IB and IO. Okay. So when I fuse this with this, what do I get? I get IA, IB. The blood group will be AB, both of them are there. When I fuse this with this, I get IA, IO. So here A is dominant, so I get A blood group. Now this with this, I get IB, IO. So here B is dominant, so I get B blood group. Here I get this, so IO, IO. So OO is there, it's a recessive thing. So OO, when it comes together, you get O blood group. Okay, now what all are the blood groups that you can find here? A, B, and O, every blood group is here. So according to this, we have B as a correct option. This came in 2021 KCET. Okay, works? Guys, I'll tell you one thing. Since I have solved so many NEET questions as well, you would love to solve NEET questions. It would definitely motivate you. After this, after solving all these KCET questions, you can try them also, because I see the questions are kind of same, but the pattern is kind of different. It might be more complicated in there for NEET, but still you can find a lot of questions there. Yeah. Now match the column 1 with column 2. Autosomal. Okay, now this time we will be talking about Turner syndrome. So if you were someone who were worried about why is ma'am not discussing about Turner syndrome, now we'll discuss it. So first thing that you need to understand is that what do you mean by autosome and what do you mean by allosome? So autosome means the chromosomes which have nothing to do with the sex of an organism. So I told you we have 23 pair of chromosome. Out of that, 22 of them, they are known as autosomes. The 23rd pair, which is a sex chromosome pair, it is known as allosome. Okay, so when I say that there is some disease that is autosomal, okay, or basically autosomal disease, that means that disease will be caused when there are certain changes in the number of chromosomes of this particular thing. Okay, when trisomy and monosomy comes. Okay, there are certain diseases which is autosomal recessive gene, autosomal dominant disease, that comes under Mendelian diseases. That means there you are talking about a chromosome. In the chromosome, in the chromosome which is an autosome, okay, which has nothing to do with the sex of organism, in that there is one gene that is getting affected. That is what you mean by autosome recessive, autosome dominant thing. That is something different. Over here, when you say that autosomal trisomy is there, that means in the autosomes, there will be one extra chromosome somewhere. Trisomy as in you have already a pair, and in that pair you have one more coming. That is autosomal trisomy. Now, if you say autosomal monosomy, that means probably you should be having a pair, but instead of a pair, you are just having one chromosome over here. So, whenever there is something that has to do with autosomes, you call it autosomal diseases. Now, in the sex chromosome, for example, you saw in Klinefelter syndrome, you have 2X chromosome coming from a parent and one Y chromosome. Over here, the allosome should be actually 2, but you have 3 now. So, this is a disease that is caused by allosomal thing. So, when allosomal trisomy means, for example, 2 of the normal chromosome should be there, but extra 1 chromosome is coming, that means it is allosomal trisomy. Okay, what do you mean by allosomal monosomy? That means it should be actually 2 in number, but only 1 is there. That means in the allosomes which should be 2, there is only 1 coming. That means you are talking about allosomal monosomy. Okay, now a little thingy for you. Down syndrome, you tell me, okay? Down syndrome, I say it is caused when the chromosome number 21 has one more copy. Chromosome number 21 has one more thing. So, I say it's a trisomy condition. Okay, so usually it should be a pair of chromosome, that is a pair of 21st chromosome, but you have one more chromosome coming around, which is why you get the disease that is Down syndrome. It is is it autosomal or it is allosomal? Is it autosomal or it is allosomal? Children, 21st chromosome, remember until 22 all of them are autosomes. They do not have to do anything with the sex of the organism, right? 21st chromosome, that means it is autosomal. So, Down syndrome is basically an autosomal disease, and this is autosomal what, children? Trisomy. Okay, when you talk about Klinefelter, syndrome, over here you say that basically you should be having XY as a chromosome number, but you are having XXY. Aren't you talking about sex chromosomes? Yes, and sex chromosome, what do you call them? Allosomes. What do you call? Allosome. Now, you have an extra X chromosome. It should be a pair, but you have an extra pair. So, what do you call extra number? What do you call it? Trisomy. So, this is allosomal trisomy disease that you are having in the case of Klinefelter syndrome. Now, talking about Turner syndrome. In Turner syndrome, what happens is that usually you should be having XX chromosome, but in them, you're losing this. There is no X chromosome coming from one parent. So, basically, it will be just one single X chromosome. So, you should be having a pair, but you are not having. Again, this is happening in sex chromosome. So, it should be allosomal disease. And here, you are deleting one. You are losing one chromosome, one sex chromosome. It's just XO condition, where there is no other sex chromosome. There is only one sex chromosome. So, this Turner syndrome occurs in female. The person is female, but would be having male characters. But would be having male character as well, then they will be sterile. They won't be able to make babies. They won't be fertile. They would be sterile, my dear children. Okay, so this is XO condition that is occurring over here. It is allosomal disease. And is it trisomy, children? No, you are not gaining an extra chromosome. Okay, you are losing a chromosome, which is why it is known as monosomy. There is only one chromosome for this. This is allosomal monosomy disorder. Got it? So, now let's see. Okay, now let's see. So, you have allosomal trisomy. That becomes very easy for finding things out. For allosomal trisomy, you have Klinefelters. At least this thing you know directly. Okay. Allosomal monosomy, you know it's Turner syndrome. Autosomal, there is only one autosomal disease that I can see over here, that is Down syndrome. Cystic fibrosis, which is left, does not match any of this category, goes into Mendelian disorder, because here you are not talking about the number of chromosome, it's the defect in a gene in one particular chromosome. So, this becomes your answer. So, autosomal, so A for 4. A for 4, you have two options. So, let me eliminate both these options. Okay, now let's find B. B is allosomal trisomy and it goes with Klinefelters, so it is to be 3. So both this option again are correct. C, if you see allosomal monosomy, it should be Turner syndrome. So she should be with 1. So then this gets cancelled and you have this as the answer. Okay, KCET 2021 question. Next question. Which among the following characters selected by the Mendel in a pea plant is a recessive character? So you know he actually chose 7 pair of contrasting traits. He chose 7 pair of contrasting traits. For example, stem height, seed color, pod shape, seed shape, flower position, flower color, lot of things were there. So in that they are asking you what is a recessive character. Okay, you have in all of them, for example, when you talk about stem height, you have 2 things. One is tall and the other one is dwarf. Which one is dominant? Tall is dominant. Which one is recessive? Dwarf is recessive. So they are asking you which among these things comes under recessive characters that he chose. Okay, inflated pod, when the pod is like full. The green color thing that you see, no, the covering, if it is full, it is known as inflated. So inflated pod is an example of dominant trait, whereas constricted one, the one that is so dry and thingy, that is the recessive trait. Okay, now green pod color, that means if the color of that whole thing, the outer covering is green, is it dominant or recessive? It is a dominant trait. Green, we also go to shops and we search for green ones. No, do we choose for yellow ones? No. So the yellow one, which is the opposite one, that is recessive. Usually we have green. Now white flower, is it the white flower which is dominant? No. Violet flower of pea plant is dominant. White flower is recessive trait. Now this is a recessive trait. How can you remember this, ma'am? I do not have any option. We have to remember it anyhow, seeing this thing that you, what do you call it? The chart. Okay, I have one way. Gravity. Okay, gravity stands for all the dominant traits. But you have to know which one. Green, for example, G for green, but you should be knowing what is green, pod color or it's something else. So that is the thing, that is why I am not teaching you this. Okay, so please take the reference of your book. You have these things over here, all these are the recessive traits and these are the dominant traits that you are having. You can use gravity as a thing where G stands for this, R stands for this, A stands for this. You can go like that also if you want. Okay. Now axillary flower means flower which comes off like at the axillary position. So this is a dominant trait, yes. Terminal flower, that means flower that comes over here, that is a recessive trait. So only the white flower which is there, this is a recessive trait. This came in 2021. Okay, next. Which of the following characters was not studied by Mendel in his pea plant experiment? Leaf shape, stem height, pod shape, seed shape. It is leaf shape. He had nothing to do with it. This came in 2020. Okay, easy question, direct question. In an organism, mutation in a single gene exhibits multiple phenotypic expression. Again, they are talking about the same thing. Okay, where I told you one gene is basically going to control trait 1, trait 2, trait 3. One particular gene is doing all this. So in an organism, mutation in a single gene is exhibiting multiple phenotypic expression, as in multiple changes, multiple phenotypes, multiple character. Identify the underlying genetic mechanism in the above instance. What do you call this? This is known as pleiotropy. This is known as pleiotropy, my dear children. Okay, I will be explaining you the example of pleiotropy also if the question comes. If not, I will just explain it to you. Okay, 2021 question. Multiple allelism is when you have multiple alleles for a single gene. For example, for blood group, you have A, IA, IB, IO. You have multiple alleles. That is what you call by, what do you mean by multiple allelism? Incomplete dominance, as in, for example, I have capital R, I have small r. Usually, this should be showing up its character. But over here, for example, usually it should be like this and it should be red in color, because R, capital R stands for red and small r stands for white. Okay, so it should be red, but here red is not being shown. Instead, pink color is shown. So it is intermediate thing that is being formed between red and white. So that means it is not completely dominant, it is incomplete dominance that is shown in snapdragon flowers. Okay, polygenic inheritance basically means for one trait. Usually we said that one gene codes for one trait, but we are saying a trait is controlled by multiple genes. For example, gene 1 is also contributing to this, gene 2, gene 3. So polygenic as in so many genes are coming together to bring out one particular trait. Example is the skin color. So you have multiple genes for your skin color, for coding your skin color. For example, this guy is not producing much, that means your skin color will go down. So things like that. Okay, so here you have pleiotropy as an answer, 2021 question. Going ahead, a pure breeding pea plant with round yellow seeds was crossed with pea plant having wrinkled green seeds. Okay, pure breeding, that means it has to be capital R, round and yellow. Pure breed means you're talking about homozygous, and it was crossed with what? Pea plant having wrinkled green. So it is small r, small r and small y. On selfing of F1 hybrid of this cross, 64 progenies were obtained in F2's generation. Find out the number of F2 progenies showing non-parental characters. What are you talking about? Do you know dihybrid cross? Yes, we know dihybrid cross. So when you cross this, you get all of them to be capital R, capital Y, small y. This is round yellow, but they are heterozygous in condition, you see. Okay, and now when you self them children, you get something like this, 9:3:3:1. Okay, where 9 ones would be round and yellow. They are the dominant. This is the dominant trait that we begin with. Okay, round and yellow, this is the dominant trait. 1 will be the recessive trait, so it will be wrinkled and green. Now 3 and 3 which are there over here, this will be standing for different things. Okay, different combinations that was not seen in the parental generation. This will be the new combination that you could be finding. So round and yellow is there. Let's make round and green. So 3 stands for round and green, and here wrinkled and green was there in the parental generation, but now you would be having wrinkled and yellow. So these are the new combinations that you are finding over here. And what does the question say? Find out the number of F2 progenies showing non-parental character. So you have to find the exact number of progenies which are going to show this character, non-parental. Okay, these are both parental character. This is non-parental, and you are given the total number of progenies. Total number of progenies when you did this experiment was 64. So how will you find this non-parental combination, my dear children? You should be knowing this ratio. 9 plus 3 is 10, 11, 12, 13, 14, 15 and 16. Total it. So you have 64. 16x is equals to 64. This would be the mechanism. Okay. Now when you calculate it, it becomes 4. X is equals to 4. X is equals to what? 4. Now what you have to do? You have to multiply things. So 9 into 4, 3 into 4, 3 into 4, 1 into 4. So when you do this, you get what? 36, 12, 12 and 4. Together if you calculate, it will be 64 only. Okay, it will be 64. Now we are asking about the number of organism or progeny showing non-parental characters. So non-parental are these both. So you plus this 12 plus this, you get 24 of them. The answer becomes 24. This came in 2020, and for knowing this question, you should be knowing this. So whenever you're talking about dihybrid cross, my dear children, without going through the entire question, you don't have to invest too much time into it. Just understand you're going to talk about this particular ratio. So you should be knowing this ratio. If you're messing up with this, and if you're not knowing that 9 stands for round yellow, and 4 basically the 1 stands for the wrinkled and green. This is a recessive parental trait. This is a dominant parental trait. Okay, both of them are parental. Rest in between, you have new combinations. So you should be knowing about that. Then only you will be solving this question. Works? Next question. 17. Identify the odd one among the following disorders: hemophilia, sickle cell anemia, phenylketonuria, thalassemia. So I'll say phenylketonuria. Okay, why, children? Hemophilia is a blood disorder where the clotting enzyme is not being produced, hence the clotting won't be happening. If, for example, I get a cut, the blood will go on. Sickle cell anemia is where my hemoglobin chain, which is beta hemoglobin, it is not properly produced. It's a defective version of beta globin which is produced, which is why the cells will be sickle shell in shape, the blood cells, the RBCs. Thalassemia is when again my alpha and beta globin chains could be affected, which is why the right amount of hemoglobin chains won't be produced. Okay, and that causes whole trouble. Phenylketonuria is a disease which happens, and this is an example of pleiotropy. I told you pleiotropy. What happens exactly over here? So in our body we have a amino acid which is phenylalanine. And this phenylalanine, no, it is converted to something that is known as tyrosine with the help of one particular enzyme whose name is phenylalanine hydroxylase. Phenylalanine hydroxylase. Fine, that is the name of the enzyme. Now, I want you to understand that this particular enzyme is coded by one gene. This particular enzyme is coded by one gene. Now, this enzyme is not produced. So, what will happen? Basically, this whole process would not be happening. Instead, this phenylalanine will be converting itself into phenyl pyruvic acid. And it is stored in our blood, brain and things like that, making us mentally retarded. That is the problem that is being caused by this. Now, I am going to tell you one more thing. In pleiotropy, if you remember, one gene is not responsible for just one trait. Instead, it is responsible for multiple characters. So, over here, since this phenylalanine hydroxylase is not being produced, the gene is not exactly coding for it, there is some mutation over here, which is why obviously tyrosine is not being produced. But apart from that, your skin and hair pigmentation is also affected. Okay, it will be hypopigmentation. That means it will be light in color. Why, children? Because this tyrosine, which has to be produced like this, was responsible for production of melanin also. Melanin is responsible for pigmentation. So basically, because of one enzyme not being produced, you are affecting multiple traits. Okay, so which is an example of pleiotropy also. So phenylketonuria is caused like this. This came in 2019. Cool? Next question. From the chromosomal complements given below, identify the one which shows female heterogamety. Female heterogamety. So until now we were talking about male heterogamety. XY and XO. Female heterogamety is shown by just one group of organisms, that is birds. So in birds, no, what happens? ZZ stands for males. It is for males. So this is homozygous condition and this is male homozygous. Homogamety. Whereas ZW means it's a female. So female now will be producing two type of gametes. One will have Z and the other one will have W. Get it? So this is female heterogamety that you are finding in the case of birds. 2019 question. 20th question. Sickle cell anemia is due to the following mutant gene. So sickle cell anemia is due to the following mutant gene. It is this. Again a direct question. How can you remember it? Remember GAG and GUG. GAG and GUG. So usually children get confused whether GUG was first and then it gets converted to GAG. No, please remember in alphabetic order, A comes before U. So it was A first and the A is replaced by U. So basically this codon was coding for an amino acid, that is glutamic acid. Now since it is changed to U, it will be coding for valine. Again, how will you remember this? G comes before V. So this is replaced by this. So now this will alter the beta globin chain or the beta globin protein that is going to be produced. 2018 question. Works. Next question. ABO type of blood group in man is an example of pleiotropy? No. Incomplete dominance? No. Codominance? Yes. A and B, both of them can come together and show codominance. Multiple allelism? Yes. Because you have multiple alleles, A, IB and I, that is O, right? So you have C and D, that is correct. C and D only. 2018 question. Okay. Next question. Choose the possible genotype responsible for lightest skin color in human beings. I told you you have multiple genes coming together for something, for one particular trait, that is for example skin color. Okay, so usually you think you have, when all of them they are capital, it will be giving you the darkest color that is possible. The darkest color that is possible. Now suppose small a is there, small b is there, small c is there coming, it will be giving the lightest color. So it is A, B and C like this, not like this. How can you write? So basically, I hope you know that for every gene you have alleles. For every gene you have alleles, right? So it can be capital A like this, capital B and capital D like this. So in total I can write it capital A. This will be the darkest color coding gene. Okay, over here it can be like this. Okay, where this is capital A. So here also there is a dominant thing that will dominate, and hence they will also show an intermediate color. It is not completely dominant. This is an example of incomplete dominance also. Okay, it is not completely dominant, which is why it will give you an intermediate color. Intermediate color. It is not completely dominant. I am writing incomplete dominance. Incomplete dominance. Now suppose there is AA gene, there is BB and there is CC. It gives the lightest color. It will be AA, BB, CC. Okay, so answer becomes, which one among them is going to give the lightest color? You tell me. So here there are capital things, so I will go wrong. Okay, here if you see they are all again something like that, so this also becomes wrong. So there is one capital, there is one capital, there is one capital. So they will show something that is darker color, lightest color you are asked, no? Here also if you see there are two, there is one, there is two, again something. But here there is only one dominant, rest all of them are this one, so recessive. So this becomes the answer. 2018 question. Okay, and what do you call this condition? Polygyny, where multiple genes are coding for one particular trait. Cool? Next. Both male and female have normal vision, though their fathers were colorblind and mothers did not have any gene for color blindness. The probability of their daughter becoming colorblind. So both male and female have normal vision. Understand that question. Male, female have normal vision. Okay, though their fathers were color blind and mothers did not have any gene for color blindness. So whenever this thing comes that male has normal vision, that means male is normal for sure. It should be XY, because males with one chromosome like this, or basically one mutation like this, will be color blind only. So if I am saying that they have a normal vision, that means there should not be any mutation, it will be just XY. But in the case of female, it said basically they have normal vision, but the father was colorblind. Now you only think, if the father is colorblind, okay, suppose the mother is not colorblind, the father is colorblind, over here anyhow whatever you do, this father will be contributing X, right? Like this only. Even it crosses with this, you get XCX. Even it crosses with this, you get XCS. So basically, whatever females will be produced will be a carrier for sure. So this female who is said to have a normal vision is a carrier for color blindness. You got it? So if it is said the female's father is color blind, that means the female has to be a carrier, my dear children. Whatever you do, there is no option. Okay, please understand that fact. And assuming that, okay, obviously they cannot have both the conditions like this, then she has to be colorblind. She cannot have normal vision. But in the question it is said that they have normal vision. So this should be the thing. Now when they cross, what is the probability of the daughter becoming colorblind? Let's see that. So over here I am going to cross it here. So I have XCX from mother and XY from father. When I cross this with this, I get XCX, and I cross this with this, I get XCY. When I cross this with this, I get XCX. I guess this thing. Have I did something wrong? Yes, I did something wrong. Just a second. Just give me a second. Proper things. Okay, let's begin the battle. So I have X and XC and XY over here. Okay, this is the gamete number 1, this is gamete number 2, this is gamete number 1, this is gamete number 2. When I fuse this with this, I get XX, it's a normal female. When I fuse this with this, I get XY, it's a normal male. When I fuse this with this, I get XXC, this is a carrier female. Okay, when I get this with this, I get XCY, it is colorblind male. Why colorblind male? Because there is no one to overshadow the effect. Over here if you see, there is someone to overshadow the effect of this. So this would be a carrier, this won't be a colorblind person. So if you see the females, there is no one who is colorblind. One is carrier but not colorblind. So I'll say 0% of them is basically colorblind. This came in 2018. Cool, works. If you're asked what is the probability of sun becoming colorblind, what will be the answer for that? What is the probability of the sun becoming colorblind? Can you try it? There is only one sun that is colorblind. Can you tell me in the answer? Okay, in the comment section, not in the live chat. In the comment section, what is the answer for this? 22nd, if I say it's a sun, if a sun, then what? What percentage of the sun will be colorblind? 23rd, the given disorder phenylketonuria is an example of direct question pleiotropy. I already explained you, 2016 question. Now, 24th question. A human male is heterozygous for autosomal gene A and B. Heterozygous for autosomal gene A and B. Okay, heterozygous means, for example, there is A gene, there is B gene. Heterozygous means it cannot be AA and BB. It has to be capital A, small a, capital B, small b. Okay, they are heterozygous. Now, he is hemizygous for hemophilic gene. So, hemophilia is a sex-linked recessive disorder. So, I hope you know that it will be on the sex chromosome. And this and he is hemizygous for hemophilia genes. That means what, children? Basically, he has this condition. This is there, and this is the thing that he is having. Okay, what percentage of sperms will carry ABH gene, genotype? What percentage of them will be carrying? So, instead of writing like this, just understand, you're talking about H gene is here, like this. Okay, this is the H gene. So, what percentage of them, the sperm will be carrying ABH gene? So, what all does he have already? He has AA, BB, and he has this. Okay, let's make combination. So, first combination is either this gamete, either this chromosome can come, this chromosome can come, or this chromosome can come. So, it can be A, capital B, and XH. Okay, next, capital A can come, small b can come, and XH can come. Agreed? Next thing that I can have. Okay, so from here I can have small a, I can have capital B, and I can have XH. That is one combination. Okay, next thing that I can find is what? I can find, wait, am I messing around? Wait. Sometimes things happen which should not be happening. Okay. Same thing I can do with Y also. So, it is A, capital B, and it can be Y gene. Similarly, it can be again A, small b, and it can be Y. Cool? Works. So, you have 4 combinations over here. Now, I'll go with the small letters. So, it can be small a, it can be capital B, and it can be XH. Okay. Next thing, it can be small a, it can be small b, and it can be XH. And next thing, it can be small a, it can be capital B and Y. It can be small a, it can be capital B, and it can be Y. Now, the question is asking what percentage of the sperm. So, this is the type of sperms that could be made, okay, type of the genotypes that could be found in the sperm. What percentage of the sperm will be carrying ABH? So, ABH means it should be looking like ABXH. Let's see. If you see from here, okay, we just have this. We just have this which is carrying ABH gene. Got it? Did you get it, my dear children? So, how many? 1, 2, 3, 4. 1, 2, 3, 4. So, almost 0.25. That means 25% of them will be having this particular gene. Get it? So, this time you have to use a little bit of a brain. Okay, you might get confused in the whole process, but let's just start from here. This, and this, then this, and this, then with the next one. This, and this, then this, and this. When you do this, just make it with Y also. When you do with this, just make it with x and then again with y. You will be able to find it. Now 8 combinations are possible, so 1 by 8 will be 0.25. Fine. Next question. From the following pedigree chart of a family, one can make an analysis of what? What type of disease it is? Autosomal dominant trait, autosomal recessive trait, allosomal dominant trait, allosomal recessive trait. So if it is allosomal, then allosomal means sex linked. So basically all the sexes should be having the disease. So I will be cutting it off because I don't see that. Here I see one male is there, one female is there. This is not affected, and you get three males, and that one male is only affected. So I cannot say that is allosomal. So I'll cut that off. Now it is autosomal dominant trait or autosomal recessive trait. That is the next question that I am having. If it is a dominant trait, then it should be shown in all, but it is not shown in all. So it has to be a recessive trait, my dear children. Okay, if you see, it is not shown in all the progenies over here. If it was dominant, it would have been shown somehow. Cool? Works? So this is the answer for 25th question. Going ahead, 26th, double lines in a pedigree analysis shows. For example, when you see this line, what does it show? That is consanguineous marriage, that means marriage between relatives, my dear children. This is a direct question, came in 2015. Next question, which of the following condition correctly describes the manner of determining the sex in the given example? Okay, homozygous sex chromosome XX produce male in Drosophila. Is it male? I told you Drosophila and human beings had the same thing. So XX is for females. So this is wrong. XO type of sex determination, male sex, determines male sex in grasshopper. Grasshopper is an insect, and insect has XO type of sex determination, where XO is basically for males. This is correct. Next, homozygous sex chromosome ZZ determines female. No, this is the only place where female are heterozygous. So they are ZW in birds. So this is wrong. XO condition in humans as found in Klinefelter syndrome. No, it is found in Turner syndrome. In Klinefelter's there is XXY. So this is again wrong. So this becomes the answer, my dear children. This came in 2015. Okay. Next question. Statements A. Statement A. For a particular character in an individual, each gamete gets only one allele. For a particular character in an individual, each gamete gets only one allele. Correct. For example, capital T, small t. In the gamete, there will be either one, this one. So it can be either small t or it can be capital, not both. So this is correct. Chromatids of a chromosome split and move towards opposite poles during anaphase of mitosis. So chromatids basically, for example, if you see this, children, these are the chromatids. This is one, this is second. Those are split and they go to the opposite poles during anaphase. Yes. During anaphase, they are getting separated and they move to the opposite poles. That is also correct. So you have different phases: metaphase, prophase, metaphase, anaphase, telophase. So prophase basically means they are scattered over there. Metaphase basically means, if for example this is a circle, everyone is aligned over here in the center. Okay, this is metaphase. Anaphase means they are now getting separated. Telophase means they have reached the region and now you have different things being formed around. So statement 1 and statement B both are correct, but it does not explain. There is no explanation of both of them. Okay, for a particular character in an individual, each gamete gets only one allele. Actually it is correct. Actually it is correct. Let's see. So statement A is correct and statement B is wrong. Both the statements are correct and B is the reason for A. Statement B is correct and statement A is wrong. Both the statements are correct and B is not the reason for A. The reason spelling is wrong. Okay, I'm getting confused, but I suppose this is the answer, but I'll just confirm it. Okay, this came in 2014. Yes. Next, sickle cell anemia is caused due to substitution of what? Sickle cell anemia, I told you, GAG converts to GUG. A is taken by U, and over glutamic acid is now not being formed, valine is being formed. So valine at the 6th position of beta globin chain by glutamine, no. Valine at 6th position of alpha globin chain by glutamic acid, no. Okay, first thing, glutamine is something different, it's wrong. Okay, valine at 6th position is correct. Okay, alpha globin chain is wrong. Glycine at 6th position, that is also wrong. Glutamic acid at 6th position of beta globin chain by valine, this is correct. So glutamic acid is changed by valine, replaced by valine at the 6th position of beta globin chain. It's not alpha, it's beta globin chain. Okay, 2014 question. Now 30th question, Down syndrome is an example for what? Syndrome caused in allosomic mutation. Allosomic, no, it is autosomal, chromosome number 21. Aneuploidy of sex chromosome, again not sex chromosome. Loss of one sex chromosome from the diploid set, no. Aneuploidy of autosomes, this is correct. There is a gain in one autosome. 2014 question, and with this we are going to end the number of questions. So I hope that each and everything that I taught you now is understandable to you. If there is any change, I'll alter it, I'll make it correct. Do not please be worried about it. Okay, so that's all, my dear champions. If you think that this particular video was definitely helpful for you, do not forget to comment down your valuable comments. Would love to see that. Okay, and also study. Do not forget to do that, and do not be panicked. Everything is going to be really, really good. And do not forget to subscribe to our channel, that is Deeksha Karnataka. Bye bye. Take care.